Welcome to Genetics Unsealed, the podcast where we unravel the mysteries of biology, genetics, and evolution. I am Fulby Melishni Odeleke, and today we are diving into a fascinating topic that connects all life on Earth, our universal genetic code. Despite the incredible diversity of living organisms, from the cyanobacterium to the majestic blue whale, nearly every life form follows the same genetic instructions. But how did this universal code come to be? And could there have been other genetic codes before ours that are now lost to time? Join on this journey. We will also explore how this genetic code influences embryonic development, shaping organisms from a single fertilized cell into complex beings. So whether you're an A-level student here for revision, a scientist, or just a curious mind, stay tuned. Okay. The story of the genetic code takes us back billions of years ago to the origins of life. A recent study led by Source and Webby, a doctoral student at the University of Arizona, has challenged the conventional understanding of how the genetic code evolved. Published in the journal PNAS, her research suggests the order in which amino acids, the building block of proteins, were incorporated into life's code may not be what we previously thought. Now let's break this down. The genetic code is essentially a biological dictionary. It translates sequences of 4 nucleotides, adenine, thymine in DNA, or uracil RNA, cytosine and guanine, into proteins made of 20 different amino acids. But these amino acids didn't all arrive at once. Life's earliest forms likely use a similar set of amino acids, and more complex ones were added over time. Webby's study found that early life preferred small amino acids, whilst those that could bind to metals, crucial for biochemical reactions, were integrated much earlier than expected. This contradicts the widely accepted idea based on the famous 1952 Urey-Miller experiment, which attempted to simulate the chemical conditions of early Earth. While the experiment proved that simple organic molecules can form naturally, it did not account for sulfur-rich amino acids, despite sulfur being abundant on early Earth. This emission suggests that sulfur-containing amino acids may have been present much earlier than previously assumed. Even more intriguingly, Webby's research hints that today's genetic code might not have been the first. Earlier, now extinct genetic codes may have existed before life is settled on the universal one we use today. This raises profound questions. What alternative genetic codes were used by life forms that no longer exist? And could alien life, if it exists, follow a completely different biochemical code? Now, switch on to a different topic. Embryonic development. The same genetic code that governs all of life also plays a crucial role in how complex organisms form a single cell. One of the most remarkable discoveries in genetics was the identification of Homeobox genes, which are control genes that regulate the development of body structures. Homeobox genes are highly conserved across species, meaning they have remained virtually unchanged for millions of years. They are grouped into clusters, known as Hox clusters, which are responsible for determining an organism's body plan, where the head, limbs, and internal organs develop. These genes are activated in a specific sequence from anterior to posterior, ensuring that the structure is formed in the correct locations. For example, Hox genes in vertebrates determine where limbs grow. If these genes are mutated or disrupted, developmental abnormalities can occur. The fact that fruit flies, humans, and even fish share similar Hox genes suggests that this genetic blueprint dates back to our early common ancestors. But development isn't just about growing, it's also about refinement. This is where apoptosis, or programmed cell death, comes in. Apoptosis is a highly regulated process that ensures the proper formation of structures. Human fingers, as an example. During early development, our hands initially form as web structures. Apoptosis removes the unnecessary cells between the fingers, allowing them to separate. Without this process, we'll be born with webbed fingers and feet, much like amphibians. Apoptosis also plays a crucial role in the immune system, eliminating ineffective or self-reactive T lymphocytes, which cells that help defend against disease. Without this process, our immune system can mistakenly attack our own tissues, leading to an autoimmune disease. The mechanism of apoptosis involves a cascade of signals often triggered by molecules like nitric oxide, which initiate the breakdown of the cell structure. Enzymes dismantle the cytoskeleton, DNA fragments into pieces, and the cell breaks apart into smaller vesicles, which are quickly engulfed by immune cells. This ensures that no harmful debris is left behind. The discovery that life's genetic code may have evolved in a different order than previously thought has profound implications. Could we one day reconstruct extinct genetic codes? What does this mean for synthetic biology, where scientists attempt to design artificial life? And if life were to evolve on another planet, will it follow a similar genetic system, or will we encounter something entirely alien? Additionally, understanding the genetic mechanisms behind embryo development could revolutionize medicine, stem cell research, regenerative medicine, and genetic engineering. Genetic engineering all rely on deciphering these fundamental biological processes. Imagine a future where we could repair damaged tissues using precise genetic control, or even design new life forms with entirely novel genetic codes. From this takeaway, genetics is beautiful. Never forget that. Thank you for tuning in to Genetic Unsealed. This is your host, Premier Leshlo Delegate, signing out.